Hi everyone, this is Mr. Hall and this is a video about factors that affect enzymes. Previously I've talked about the required lab you need to know about enzymes and how it's affected by temperature, pH, and I've talked more generally about enzymes before. This is more talking about the things that can affect how active those enzymes are in your digestive system as well as elsewhere. So the objectives for today's lesson are to describe the ideal conditions for different enzymes to work in the body and also describe and explain what would happen to enzymes in different conditions. So first off, all enzymes have a thing called an active site. And this active site is where the substrate, which is basically just a fancy word for the food molecule, it fits onto the enzyme and where the reaction happens before the products then break away. So the food attaches onto it, there's a chemical reaction, it breaks off, and then the food is broken down into a smaller piece as a result. The enzyme remains unchanged. Now, this active site on the enzyme is a very sort of sophisticated structure. So this image, which I quite like, shows the 3D nature of enzymes, which are very complicated structures, you know, in a 3D sense. So the food substrate, or this food bit, has to kind of fit in perfectly into this area in order to have the chemical reaction happen. Now, that looks quite complicated, so often we show it as a 2D structure. So if you think about it like this structure here on the left-hand side, this is my enzyme, okay? And on the right, this is my reactant or my substrate. And basically what that is your food molecule. So let's say, for example, this is a carbohydrate like starch, okay? And it comes inside and you can see it fits in perfectly into this space, and this space is your active site. So they come together like so, and then they connect, okay? And it's sort of, it can go both ways. It's sort of an equilibrium, which I won't go into much detail now. So they slot in perfectly like so. There's a chemical reaction where this reactant, this substrate, gets broken down into two different things. And then after that, it then forms two products. And this is basically your food molecule broken down to smaller pieces, which moves away. So this is your product. And notice that your enzyme stays exactly as it was at the start. So you've got an enzyme and a reactant at the beginning. You then move on to your enzyme reactant complex, which is like an intermediary stage between the beginning and end of the reaction. And then you've got your enzyme and your products, which go away. So now this active site, as I said, it has a specific shape. And you'll notice from this animation I showed you is that the substrate here fits into the active site of the enzyme absolutely perfectly, okay, like a key fitting into a lock. That's why this is often called a lock and key mechanism, where the lock is the enzyme, which is here, or if you could say the lock is the active site on the enzyme, and then the key is your substrate, which fits in, all right? So that's why it's called lock and key, and it's quite a useful way of thinking about it. Now, enzymes can only work with particular substrates. They're not going to work with any substrate. For example, I'll come to an example in a minute actually, but the reason for that is because there are different shapes between the enzyme as well as the substrate. So for example, if we think about carbohydrates, which is an enzyme, they'll have a very specifically shaped active site, and it can only break carbohydrates because those shapes fit into each other. Whereas proteins or lipids, or fats rather, or lipids, they can't be broken down by carbohydrates because the shapes are different. So for example, if we look at these two, here we've got carbohydrates, which is this enzyme, and you've got carbohydrates, which is over here, such as starch, and they fit in perfectly, as you can see. However, if I try and do carbohydrates and protein, you can see that the shapes are different, okay? On protein, you've got this kind of triangular shape and a square shape, so they're not going to work. Here, I've got this kind of square and sort of got a shaved off bit here and a round part there, which isn't going to work as well. So the lock and key mechanism is a really useful way of thinking about how specific enzymes only work on specific substrates. Now, this reaction between the enzyme and the substrate, it has a particular speed, and we often call it the rate. Okay, and later on I'll do a video about the rate of digestion and some calculations with it. Now that speed can depend on several factors. Now the two most important that you'll probably come across are the temperature and the pH, which I'll go into in more depth later on. But there are other things that can affect it as well, which you need to be aware of. So the enzyme concentration. So if you have a solution containing an enzyme, if there's lots of enzyme per unit volume, it's going to work a lot quicker at breaking down your substrate. It depends on the food concentration. So if you've got lots of food concentrated in a solution, it will break down quicker. It depends on the surface area of the food. So a block of carbohydrate, for example, will probably be digested a lot slower than individual bits broken down. And also pressure, that's more to do with gases really. Now, all enzymes work best at a particular temperature and a particular pH, and that's called the optimum. And different enzymes have different optimum temperatures and different pH values at which they work best. Now, if you change the temperature and the pH significantly beyond the optimum, the shape of the enzyme changes. And if the shape of the enzyme changes, it changes the active site shape. And that means that the enzyme can't work anymore because the substrate can't fit in to the enzyme's active site. When that happens, the enzyme is what we call denatured, and that means that the shape has changed to the point where it won't work anymore. And this is a good example. So if you look at this enzyme, it's normal. If I heat it up too much, or if I change the pH too much, either plus or minus, it changes the shape, and you can see this active site has now got a dramatically different shape. So this substrate which I had before, which looked a little bit like this, it looked a little bit like that, it'll no longer fit in because this sort of round area won't fit in with the slightly changed shape, and this square bit won't fit into this jagged shape. So that no longer works. So if your enzyme is denatured, your enzyme activity won't work. And that's partly one of the reasons why if you have a temperature, it can be quite dangerous because your enzymes start to denature in your body. Now, the optimum temperature for most enzymes is 37 degrees. Now, at lower temperatures, they work slowly. At higher temperatures, they tend to denature and don't work. So a temperature profile you often get is, if you look at this axis, I've got temperature on the x-axis and rate of reaction on the y-axis. So the curve you tend to get is something that looks a little bit like this. It looks a little bit like so. Whereas this is the optimum because this is the highest rate of reaction, and that tends to be around 37 degrees. Now, it depends on certain enzymes. Some enzymes work at a slightly higher temperature, some at slightly lower, but broadly speaking, it's around 37 degrees, which is incidentally the temperature of the human body inside. Okay, over here, where it's quite slow, this is because you have a low rate of reaction. That's because it's quite slow, and that kind of harks back to what you've probably learned in chemistry. Over here, you can see the rate drops off dramatically. Sometimes it's even more dramatic. It's more like this, really. It really falls off, and that's due to the enzyme denaturing. Okay, so that's the curves you get. For pH, it depends on the type of enzyme you've got and where it works. So typically what you'll have is, if we've got pH of 0 here, 7 here, and 14 over here, there are basically three types of curve you're going to get. One of them looks a little bit like this. One of them looks like this. And one of them will look like this. Okay, so basically you've got three different conditions that these three enzymes can work at. This one likes acidic conditions. This one likes alkaline conditions. And this one tends to like neutral conditions. Now, this is a very broad stroke, and at A level and beyond, you'll probably learn something a lot more sophisticated. But if it works best in alkaline conditions, it probably tells you that it works best in the small intestine, which means it has to be lipase. For acidic conditions, it has to be working in the stomach, which means it has to be protease. And then for the neutral one, I'll just draw an arrow over here, that tells you it can work at many parts of the body, including the mouth is one of the areas, as well as elsewhere, and that must be carbohydrates, of which amylase is an example. Okay, and that's it. So I hope that's been useful in terms of going through the different factors that affect enzyme activity, and best of luck with your studies.